much. Thanks for inviting me. It's really a pleasure to speak alongside such a fantastic lineup. I was going to start by saying, you've probably seen a slide like this before, but you definitely have because Greg included this figure in his talk earlier this morning, illustrating what's called the so-called variance of uncertain significance, or the VUS problem, which basically reflects the fact that our ability to sequence is vastly outpacing our ability to interpret that sequence variation. And so the number of variants being clinically classified as of uncertain significance is growing at a much faster rate than those that are classified as pathogenic or benign. And two complementary approaches are starting to make quite a big impact on the VUS problem. On the experimental side, you have high throughput approaches, collectively referred to as multiplexed assays of variant effect, or MAVES. And these are kinds of assays where you can measure the impacts of thousands or tens of thousands of variants within a single assay. Greg gave us some great examples of that. And then on the computational side, you have computational methods for predicting variant impacts, so-called variant effect predictors, or VEPs. And these are both similar in that they output variant effect scores that can often be interpreted as reflecting something related to the likelihood of a given variant being pathogenic or benign. And in fact, in some cases, they're so good at discriminating between known pathogenic and benign variants that they can be calibrated for use as quantitative clinical diagnostic evidence. And so we're now at a point where these experimental measurements and computational predictors are starting to be used in quite a large number of clinical variant classifications. But most currently used MAVES and VEPs are essentially insensitive to underlying molecular mechanisms. So the MAVES that tend to perform best at discriminating between pathogenic and benign variants are often growth rate assays that don't really tell us much about the mechanisms underlying those variants. Same with the computational predictors. The best performing predictors for pathogenicity prediction tend to be based on sequence and evolutionary information and are largely agnostic to underlying mechanisms. So what I want to talk about today is what I'm calling the variance of uncertain mechanism problem, which is essentially the fact that for lots of variants, we know that they're pathogenic, but we don't know how they act at a molecular level. And this is, of course, incredibly important, as we just saw in Stefan's talk. If you want to treat rare disease therapeutically, it's absolutely essential in a lot of cases to understand the underlying molecular mechanisms. But furthermore, I think that this is actually very closely related to the VUS problem, because a lot of the times variants remain unclassified. They remain of uncertain significance because our assays and our predictors do not adequately capture their mechanisms. So we're biased towards detecting certain types of pathogenic variants. And in particular, we tend to be very good at identifying variants that cause disease via a loss of function. So most known pathogenic variants are truncating variants, so nonsense or frame shift changes that introduce premature stop codons and cause, most of the time, a complete loss of protein function. So these are easy to spot. We just look for the stop codon. But the fact is that actually most current clinical variants of uncertain significance are missense variants. So they involve single amino acid changes at the protein level. But missense changes also often appear to act via loss of function mechanisms. So there's recently a sort of convergence of computational and experimental evidence suggesting that somewhere on the order of half of pathogenic missense variants are acting via protein destabilization. I think that this is probably a bit of an overestimate because of elements of ascertainment bias, where it's been easier with our experimental and computational approaches to find these destabilizing variants. But the fact is that a lot of missense variants clearly cause a loss of function related to their impacts on protein structure. But there's a tremendous diversity of molecular mechanisms beyond simple loss of function that can be caused by coding variants. And so I could give a whole lecture going through all the interesting different examples that we've worked on over the years, but I'm just going to show one here today because I think it beautifully illustrates that. So this was a collaboration we were involved in with Pleasantine Mill in Edinburgh, and they identified a number of patients with ciliopathy phenotypes, all with variants in this gene called TUB4B, which is a beta tubulin subunit that's important for cilia formation. And these patients had quite a range of clinical phenotypes, and they could be broadly split into 3 groups. So there were some variants that were PCD only. So if we look at the structural context of these variants, they tend to be where they were located at the interface between the alpha and the beta tubulin molecules. They looked like they would prevent the heterodimerization of the tubulin. And that was what was confirmed experimentally. This protein doesn't assemble into tubulin. But furthermore, that mutant protein appears to trap a tubulin folding chaperone, TBCD, and causing this competitive disruption of microtubule formation during motile cilia formation. So it's important, these are all heterozygous mutations. So even though it's just 50% of the protein being made is variant, it has a bigger impact because of this competitive effect. So another variant was associated with a broader spectrum of phenotypic impacts. It was a syndromic PCD. This occurs at the intraluminal surface of the protein near the Taxol binding site in a position known to bind other microtubule-associated proteins. And so this one, it can assemble into heterodimers, and then it gets incorporated into microtubules, but it severely impacts microtubule growth and dynamics. So it acts like a poison to the microtubule. So that protein gets in there and it just basically screws up the microtubule properties. And therefore it has a much broader impacts on both motile and sensory cilia. And then finally, there were variants associated with a sensory-only phenotype. These were located sort of between the outer surface and the intradimer interface, a completely different spot from the others. And they had the sort of mildest effect at the molecule, at the molecular level. They assembled into microtubules relatively well, and they had quite modest intermediate effects on microtubule dynamics. But importantly, it seemed that this was tolerated much better in the motile rather than the sensory cilia, suggesting that they disrupt interactions that were important specifically for the sensory cilia, given the location on the surface there. So basically, the point of this is just to illustrate that in one protein coding gene, you've got multiple different types of coding variation that are acting via very distinct molecular mechanisms that are causing this broad range of clinical phenotypes. So if we want to understand this, it's very important to understand these molecular mechanisms. And so one of the issues that we have is that there have been lots of detailed cell biology, biochemistry, animal models, teasing out the molecular mechanisms of variants. But this information is not very well captured in any single place, and there's a lot of heterogeneity in the literature when referring to these mechanisms. So a couple of years ago, we wrote this review sort of discussing this, where we presented a sort of large variety of different molecular mechanisms by which coding variants could cause disease. And we made the argument that you could basically classify essentially all pathogenic variants in proteins into one of 3 categories. So you have loss of function variants, which are acting, due to the phenotypic effect is due to a disruption of normal protein activity, destabilization or disruption of an interaction or an active site. You have gain of function. There, the disease is caused by the properties of the mutant protein, so something that the mutant is doing differently. It could be a new binding site or aggregation or increased catalytic activity. There's lots of ways that it can happen. And then finally, there's dominant negative effects, which are kind of like a hybrid between loss of function and gain of function, because the disease is due to the properties of the mutant. But what the mutant does is it blocks the activity of the wild type. So it's a net loss of function overall. And that could be due to competitive effects or co-assembly into complexes. So following this review, we started collaborating with the Gene to Phenotype project, which does very careful genomic disease-gene relationship curations to add a level of mechanistic curation to the process. So we came up with this classification scheme using our sort of top-level loss, gain of function, and a number of subcategories, more precisely capturing the different molecular impacts, like effects on stability or interactions or assembly-mediated dominant negative effects. And so this is now being incorporated into the Gene to Phenotype. You can see it on the sort of EBI resource. If you look up a lot of genes now, they have this mechanistic curation. So for this example, it's classified as dominant negative, and it's got a subcategorization as assembly-mediated dominant negative with links to the literature evidence of where this is coming from. So I think this is a great step towards starting to more formally curate this mechanistic information, that a lot of it exists out there in the literature. We just don't have it captured in any kind of single place. The other thing we've been really interested in is, can we predict these mechanisms computationally? So we started off trying to do this at the gene level. So we did a sort of text mining with human manual curation approach to get a bunch of labels for human autosomal dominant disease genes. And so we came up with nearly 1,300 dominant genes where we could classify their association with dominant negative gain of function or loss of function mechanisms in genetic disease. And then we took a bunch of protein features like structural features and functional properties and evolution and population constraint, put them together and trained simple classifier models to see how well we could recapitulate those labels using the gene level properties. And we get these scores reflecting the probability of dominant negative gain of function and loss of function effects. And it turns out we can do decently. So nothing close to perfect prediction, but it's a nice starting point where you can start to think, what is the likelihood of this gene being associated with different kinds of mechanisms? So this is all being incorporated now into Decipher, the widely used clinical variant interpretation platform, under these gene predictive scores. So you can look up your gene of interest to see its predictions and also click on whether there's literature curated evidence for this as well. So next, we wanted to bring in the properties of individual missense variants. The first predictor was just purely based on gene level properties. So we developed another metric that we call MLOF, for missense loss of function, reflecting the likelihood of a variant being associated with a set of missense variants acting via loss of function mechanisms that use information like effects on protein stability and clustering within the three-dimensional structure to define this property. And what we found is that this by itself could do decently, and then combined with our gene level models, we could do markedly improved phenotype level mechanism prediction. But what we found where this is probably the most useful is in explaining mechanistic differences in genes that are associated with distinct clinical phenotypes. So we found there's lots of genes where there's more than one disease phenotype associated with the gene, all associated with different variants that appear to be associated with remarkably different underlying molecular mechanisms. So this is one of the top hits from our screen in the paper, SMCHD1. We've got two very different clinical phenotypes. This Arinea microthalmia variants are shown in red. They have a very low MLOF score, which means a low likelihood of loss of function. They're very clustered and they have very mild effects on the structure, consistent with a gain of function or dominant negative effect, whereas these muscular dystrophy variants in blue are spread throughout the protein. They all have very highly disruptive effects on the protein structure, so highly suggestive of a loss of function mechanism for these. The other thing that we could do with this is we start to estimate the prevalence of these different molecular mechanisms. So we can take dominant disease phenotypes that are caused by missense variants and then calculate the likely mechanism for those set of variants. And when we start off looking at single phenotype genes, so these are autosomal dominant genes where there's one known disease phenotype. And what we see is that roughly half are predicted to be loss of function and about a quarter each of gain of function and dominant negative. If we looked at dominant genes with multiple dominant phenotypes, it's very similar. The gain of function proportion is a little bit higher. But what was really interesting is if we look at mixed dominant recessive genes, so these are genes where different variants are associated with both autosomal dominant and autosomal recessive phenotypes. And in those cases, the dominant disease is almost always associated with non-loss of function mechanisms, which I guess makes sense if you think that the recessive is usually going to be caused by loss of function impacts. So the important thing here, I think, is just illustrating how prevalent these non-loss of function mechanisms appear to be. I think there's often been a sort of assumption by people that most genetic disease, the overwhelming majority, is due to disrupting proteins and these, how they damage proteins and cause loss of function impacts. And what we're suggesting is that these kind of often more subtle and often harder to predict mechanisms actually are very common. And this is, really important because if you want to do decide a genetic therapy to treat a rare genetic disorder, it's absolutely essential to understand, if it's a dominant negative or a gain of function, and that toxic allele then needs to be targeted as opposed to replacing the intrinsic gene function. So finally, I want to talk about our work to try and experimentally characterize variant mechanisms at scale. So this is our own MAVE work that we've done, a deep mutational scan, which is a type of MAVE, in collaboration with Greg Kudla, who's here today. So this is on the gene ERCC2. So ERCC2 includes XPD, which is a multifunctional protein. It's got two distinct functions, one associated with DNA damage repair via nucleotide excision repair, and it's also associated with gene expression, being a subunit of the TF2H complex. And so both missense and truncating variants have been associated with recessive disease, with two distinct disease phenotypes. You have xeroderma pigmentosum and trichothiodystrophy, which are basically believed to be primarily associated with those two distinct functions of the protein. So the reason we're able to do this assay is because human ERCC2 can complement its essential yeast ortholog RAD3. So we can take a conditional knockout of the yeast of the RAD3 gene. We transform the yeast with a library of all possible single amino acid substitutions. We grow them together in a massively parallel competition experiment, and we can measure the fitness of all single amino acid substitutions within that protein. So it's just about 15,000 in total, 98% of possible single amino acid changes we measured the impacts of. So we get one of these nice variant effect maps showing the impacts of all the variants along the length of the protein. And what we could immediately see looking at this data is that our assay was reflective of transcriptional function and not DNA repair. So that's, I guess, what is important for yeast growth is restoring that transcriptional activity. We could see variants, in the ATPase domain, for example, had very little impact, whereas those affecting interactions with TF2H subunits were highly disruptive to the protein. So this might seem like a limitation of the assay, but this is exactly what we want, because now we have a mechanism selective assay. So we can compare these fitness measurements to the known pathogenic variants, which are scattered throughout the protein. And what we see is, the disease variants, XP and TTD, are both lower fitness than benign and population variants, but the TTD variants have markedly lower fitness than the XP variants overall. Which is consistent with what's generally known about the diseases being associated with those distinct biological functions. If we take those fitness scores and say, how useful are they for classifying pathogenic and benign variants, they're just sort of okay. They're worse than most computational models, which makes sense because the assay is not picking up part of the function of the protein. But where these become really useful is when we combine them with the computational predictions. So here, we're showing on two dimensions. On the vertical axis, we've got our DMS fitness scores. On the horizontal is a variant effect predictor, CPT1, which was a sort of top performing model. And what you can see is that the CPT1 gives almost perfect discrimination between pathogenic and benign variants. So all the benign variants in gray are on the one side of the plot, and all the pathogenic variants are on the other side of the plot. But the DMS starts to separate them out based upon clinical phenotype. So the TTD variants tend to be low, and the XP variants tend to be high. It's not perfect, and we really wouldn't expect it to be. This is, recessive disease. Many of the patients are compound heterozygotes who have two different variants that might be affecting different functions. Some variants have been associated with both phenotypes. But the notable thing here is that the experimental assay picks up the difference between disease phenotypes, and none of the computational predictors do anything like this. And this is not just specific to ERCC2. We've seen it in other cases. So for example, another deep mutational scan we did in PAX6 DNA binding domain a couple of years ago, also with Greg. And in that case, what we were measuring in the assay was strength of binding to DNA. And what we find there is those measurements were quite predictive of disease, but none of the computational predictors showed any correlation with severity, whereas the experimental measurements did, again, not perfect, but they were the only thing that show any discrimination on the basis of phenotype was the experimental measurements of DNA binding. So I think going forward, this is going to be a really powerful approach in terms of starting to characterize variant mechanisms at scale, using these mechanism-sensitive MAVE assays. And there's lots of ways that you can do MAVEs to measure mechanisms, lots of ways that people are doing. You can think about multimodal assays where you're measuring multiple phenotypes for the same variant, and then combining these with state-of-the-art computational models that will allow us to both better address the variance of uncertain significance problem and the variance of uncertain mechanism problem. So that's it. Thank you to my group, and in particular, thanks to Mihai, who did all the mechanism prediction work, and Hassan, who did the deep mutational scanning work on ERCC2. Thank you.